Welcome to the FENS webinar on neuroplasticity across development and aging. My name is Michala Fenskova, and I'm representative of the FENS Committee for Higher Education and Training, which organizes this webinar. Just very briefly, how we came up with this topic, it basically comes from you, the society, the neuroscience community in Europe, and we collect ideas for webinars on this website. If I manage to switch to the next slide, it doesn't work. I think it's fens.org/input, but if it doesn't work at the moment, I will try to show it. Now it's here. So it's here below, fens.org/training. So you're also welcome to submit the ideas for the seminar topics here. And now I will give the floor to Anne-Sophie Hafner, the moderator of the webinar today, to introduce our speakers and the webinar. The floor is yours, Anne. Thank you.
Thank you very much, Michaela, and I would like to thank you and the FENS Committee for Higher Education and Training for organizing this webinar. I'm really excited for the discussion today, and we're going to start with two great talks. A first talk by Jetro Tuulari, and then which will be immediately followed by a talk by Thomas Wolbers. The reason is that I want to keep all the questions for the end so we can really talk all together. The Q&A is already open, so please don't hesitate during the talks to already put all your questions. I will collect them, and then I will hope that we can go through all of them at the very end. So I don't want to waste more time, so I will now introduce Jetro Tuulari, who's our first speaker today. So Jetro obtained his PhD in 2015 from the Medical Faculty in Neurology of the University of Turku in Finland. So he's also an MD, also from the University of Turku in Finland. He did, after his PhD, a little short postdoc in the same institution, and then was a visiting fellow at the Department of Psychiatry at the University of Oxford in the UK. And since 2018 and 21, actually, he then started his own group at the University of Turku, where he's both a principal investigator but also a practicing clinician. So thank you very much for being here today. He will talk, I hope, about functional brain imaging from in utero all the way to adulthood. So going from, neonatal brain and infant and adolescent. And he uses mainly functional brain imaging to look at, parameters like network organization and maturation in the brain. So I'm quite excited to hear your talk today. Thank you. The floor is yours.
Thank you for the introduction, and wonderful afternoon to everyone. Yes, so again, I come from Turku, and those of you who don't know where Turku is, it's depicted here on the map, so southwest Finland, and I work in the FinBrain birth cohort study. It is a prospective cohort study that was established by Hasse and Linnea Carlson in 2010. And it has enrolled around 3,800 families from pregnancy. And one of the key questions in FinBrain is how early life exposures affect or associate with later brain development of the children. I lead my own group, FinBrain Neuroimaging, within FinBrain, but FinBrain is a large cohort study. So there are different principal investigators for neuropsychological development, pediatric health, and many others. And in today's presentation, I will briefly discuss how plasticity occurs during brain development, but of course, bearing in mind that plasticity is actually one of the key drivers of brain development all throughout brain development. Then I will focus in this talk on metrics and measurements that we can obtain from non-invasive neuroimaging and mainly MRI imaging. And from that work, I will introduce latest developments on brain development across the human lifespan. So this will extend from early phases to adulthood. And then I will briefly discuss plasticity in the adult brain before we then move on to the next speech about, later changes in the brain. And of course, we have to define neuroplasticity somehow. It could be defined as inherent and lifelong capability of the nervous system to adapt structure and function related to changes in the environment. And these changes can be adaptive or maladaptive in terms of behavior and health. And there are well-known mechanisms for neuroplasticity. Some key ones include neurogenesis, so formation of new neurons, synaptic plasticity, and functional reorganization. Now, we cannot easily detect all of these in MRI imaging, for example. Neurogenesis we can quantify to a degree, but of course, most of the neurogenesis in the human brain occurs very early on during development and fetal development, so it's very challenging to quantify that. And of course, we can see cell death and damage. This is one of the key things clinical radiologists are interpreting in images, for example, when they are diagnosing stroke. We are unable to capture synaptic plasticity in detail, but we get good proxy measures for myelination, and we using techniques like diffusion MRI, and we can also quantify neurotransmitter levels with techniques like PET imaging. And of course, we can quantify functional reorganization across brain areas and networks, but not readily at the cellular level. Why plasticity is so important for the developing brain, or for researchers working on developing brain, is that it is commonly assumed, and for good reasons, that the brain during development is especially plastic, and this makes the brain especially susceptible to environmental influences. Here's just one example from a really nice review by Ursula Tuuli and colleagues, and they focus here on brain development related to socioeconomical status, shorthand SES. And in this plot, you see high SES children, so children born to high SES families. And on the left-hand side in the figure, you see cortical thickness, and then age in years on the x-axis. And it clearly shows how cortical thickness initially grows and then starts to decline, and that the profiles can be different for children that are living in different environments. As a proxy here, the SES, or socioeconomical status. The dashed line depicts high SES families or children, and the profile seems different. So they would obtain their peak cortical thickness later, but would also end up on a higher level of cortical thickness later on in the development. And because brain is assumed to be very plastic during development, any environmental effects before the peak in this curve can have long-term effects all the way throughout the rest of the human lifespan. So it's very crucial to first of all understand how typical brain development advances, and we made significant breakthroughs recently in understanding that. But let's start from the very beginning and from how neuroplasticity occurs or is placed in time in the developing brain. Most of the neurogenesis occurs very early on during the first half of the pregnancy or before mid-pregnancy. And neurogenesis, of course, is accompanied by programmed cell death. And then as soon as the neurons have formed, they start form synapses. And this is also very rapid already during pregnancy and occurs to be fast in the early years of life. Now, there are different processes that are also part of brain plasticity. For example, myelination starts already during pregnancy, but is also rapid in the early years, postnatal life, and then continues decades of postnatal life. So these mechanisms or processes that are often linked to neuroplasticity occur at different time points and also last a variable amount of time during brain development. Human brain development is very fast already during prenatal development, and by mid-pregnancy, so that time when most of the neurons in the human brain have already formed, the human brain already has its overall anatomical shape, but it clearly lacks something. So the brain surface is almost completely flat at that developmental stage, and slowly towards the end of pregnancy, the sulci and gyri start to form, and to a degree that term-born infant actually has strikingly similar brains in terms of cross anatomy as compared to the adult brain. And of course, this figure is not correct in scale. Neonatal brain is around 1/3 of the adult brain volume, so it is a lot smaller than the adult brain, even though they are almost the same size here. And this has been quantified also in neuroimaging research. On the left-hand side, there's one of the results from a really nice paper from Anna Nampurete and colleagues that was published in Nature a few years ago. And they used ultrasound measurements to quantify volumetric growth during pregnancy, volumetric growth of the brain during pregnancy. And you can appreciate how fast or how steep the curve in that volumetric regression line is from early gestation to gestational week 30. And now corresponding work using MRI is depicted on the right-hand side. This is from Cyan Wilson and colleagues. And you can appreciate how closely tied gestational age and brain volumetric growth are during prenatal development. And this study used MRI imaging, so in utero MRI imaging, to quantify the brain volumes. On the y-axis here, you see it's a little bit funny units, but the brain growth from mid-pregnancy to term age, brain grows from roughly 100 millimeters all the way up to 400 milliliters. So it 4 times the size during the later parts of the pregnancy. Here's a nice illustration of the postnatal growth of the brain. And this, on the left-hand column, if you will, you see a typical example of a neonatal brain MRI. And on the middle panel, you see an example MRI from a 1-year-old, and on the right-hand side panel from a 2-year-old. You can appreciate how much the brain grows in size. And I should note that the sequences, MRI sequences that have been used are similar here, and that the brain looks a little bit different. And the myelination that occurs in the brain makes it look much adult-like already at age 1. These early first years of life are the time when all of the brain tissue compartments or parts of the brain are basically growing. But cortical growth strikingly already stops growing during early childhood. And there's a nice example from early work showing this, how already during childhood, the cortical volume starts to decrease. So the peak volume for cortical gray matter is already during childhood, and towards adolescence, the cortical gray matter decreases. And from this, more recent evidence comes from this really nice article that mapped the brain growth charts for the human lifespan. So the idea here is that we are quantifying volumes of the brain very much similar to pediatric screening growth charts for height and weight. And this study by Richard Bethlehem and colleagues gathered more than 100,000 MRI images from ages 0 to 100 years of age. Our cohort FinBrain was partly contributing to this work, so and I think these findings are very important in showing how the different parts of the brain develop or obtain their peak volume at different ages. So just looking at the graphs, cortical gray matter volume obtains peak volume already during childhood. The white matter volume peak is observed in early adulthood around age 30 years, and the subcortical gray matter nuclei obtain peak volumes towards the late teenage years. So in summary, brain has a very speedy initial growth, and up to 80% of the total brain volume is already achieved by age 3 years. Most of the cortical gray matter growth stops already during childhood. Sorry, there's a banner. Subcortical nuclei grow until the age of 16 to 18 years of age, and white matter growth continues until 27 to 30 years of age. And after these peak volumes have been achieved, older ages are accompanied by initially slow decline in brain volume that then increases towards elderly ages. One important note to make here is that the brain does not develop and grow like a balloon equally on all sides. It has well-known developmental gradients that are actually nicely captured here on the left-hand side figure. This is taken from the Brain Charts article, and in the grayish colors you see the brain areas that obtain their peak volumes the first around age 6, and in reddish colors the brain areas that obtain their peak volumes later towards the age of 9 and 10 years of age. More recent work has also used similar measures, creating growth charts for diffusion tensor imaging. I'm sorry, I seem to be missing the reference here, but this is from a preprint from Michael Kim and colleagues who have used gathered data set of tens of thousands of diffusion tensor MRI images and created similar brain charts for diffusion MRI. This is maybe a few notes that are very interesting in this work is that the volumetric growth charts show, so here's brain volume, it shows a clear difference between males and females. So males are depicted in blue color and females in red. Whereas the similar differences between males and females are less clear and apparent in diffusion tensor imaging. And of course, this has also been recently done for functional MRI imaging. Here is just one plot from a recent publication, and the red colors here indicate high growth rates or high rates of functional organization development that is linked to functional brain development. It nicely captures the general trend of more red colors before the age of 6, some reddish around age of 10, and then stabilizing or decreasing rate of development towards the adult ages. What is also noteworthy here is that the somatosensory and visual areas in the brain are already or show less growth rates, meaning that they are more adult-like already soon after birth. And of course, much of this variability in brain structure and development throughout the human lifespan is also influenced by genetic makeup, but of course, genes and environment jointly affect brain development. Here's just one more illustration showing nicely how at age 7 already brain growth is already decreasing in the cortex. The white matter is still developing. At age 15, the white matter development slows already and then stabilizes towards the adult ages. And we have to stop here and remind ourselves that these are, of course, now basically mapping for each of these tissue compartments, cortical gray matter or the white matter, most rapid basis of development. So tentatively, whenever there's growth, there's increased plasticity and also increased sensitivity to environmental cues. But of course, even though these adult ages seems fairly stable in terms of the overall development across the lifespan, most of the plasticity, many plasticity studies have been done in adults. And of course, this stability makes adult participants good research participants in the first place, because we can trust that most of the variance or experimental manipulation is because of that manipulation and is not influenced by pace of brain development at that point. I was thinking about examples of adult brain plasticity, and my PhD project came into mind. In this work, we studied morbidly obese individuals. And we scanned them. So we scanned non-obese individuals, morbidly obese individuals before weight loss surgery, and also after weight loss surgery. And what we saw in this study was a decreased level of mu opioid receptors in the morbidly obese individuals before surgery, and then an increase in the mu opioid receptor levels, very close to what we observed in the non-obese individuals in the beginning, and saw no similar plasticity in the dopamine 2 receptor levels in the brain. And sort of as a conclusion, it is very important to map out these typical patterns of brain development so that we can interpret applied research studies. For example, if we have a group of participants with the clinical conditions and healthy controls, this helps us to basically interpret whether or not, let's say, a positive difference between the groups is a positive thing, indicating better or healthier brain development, or vice versa. So, for example, for neonatal studies, it is very common to interpret that higher levels of brain development or brain measures are indicative of better brain development. And of course, it's nice, again, this is from the review looking through effects of SCS. So the dashed line again kind of indicating better environment for brain development, and y-axis functional segregation. So index here would be a higher level of brain development, and the pattern can be complex. So the high SCS participant could show initially higher metrics in brain development, then have a protracted or slowed down trajectory towards the adult level, and still obtain higher levels as adults. So it is a key question to many of the developmental neuroscience studies. Is this brain development accelerated or decelerated, or can it be both? So timing and tempo of plasticity during brain development are both relevant. I thank you for your attention. I would also like to thank the funders and my research group that I'm happy to share this work with. Thank you.
Wow, thank you very much, Jetro. That was a great talk. We already have one question in the Q&A, and I hope that we have more coming. Please don't hesitate. Keep putting your questions in. As I said at the beginning, we will answer questions at the very end of the talk of Thomas Wolbers now. So I will, without further ado, move forward so that we have enough time at the end. So yeah, now we have a talk by Professor Thomas Wolbers. So he studied psychology at the university in Hamburg, and he got his PhD in 2005, and then moved to UC Santa Barbara for a postdoc where he focused on more cognitive neuroscience. And he's now a professor of aging, cognition, and technology at the German Center for Neurodegenerative Diseases, DZNE. And for over now 2 decades, he's been researching on spatial cognition, aging, and neurodegeneration. And he uses immersive VR, high-field MRI, PET, and computational modeling to look at the identity of circuit-level mechanisms. And I think for me, like recently, one of these work that I find quite fascinating is that he looks at older adults' brain, and he found that, you know, what seems to be going on during aging is more than a global performance drop, really a specific path integration errors that start to occur. And I hope that we get to discuss about that at the very end. So now, after we saw the brain developing from a neonatal or even in utero brain to the adulthood, we're going to switch to adult and aging brain. So thank you very much, Thomas. The floor is yours.
All right. Thank you very much, Anne-Sophie. Can you see the slide?
Yes.
Okay, perfect. All right. Yeah, Good afternoon, everybody. I'm glad that there's so much interest in these presentations. And I also like to thank Jetro for a fantastic introduction into mechanisms of neuroplasticity. So I don't have to explain this in detail. So as Anne-Sophie said, my group, we're working mainly in aging, in old age, and we have a bit more narrow focus on mechanisms of spatial cognition, spatial navigation, because it's a pretty fascinating function. So when you look at cognitive abilities as they develop over the lifespan, and we can look at things as you can see here, like reasoning, verbal memory, you see that, try to get the laser pointer here. There you go. So you see that many of those functions actually already start to show some decline around, say, mid-40s, early 50s, and then often the decline accelerates. But there are also some cognitive abilities, like in this case here, numeric abilities or verbal abilities that seem to be relatively stable across the lifespan, and you don't really typically see much change even in very old age. As I said, we are particularly interested in one component of cognition, which is navigation. It actually goes back to Charles Darwin, who already in 1873 postulated that there should be kind of a part of the brain that's specialized for the function of direction, because he observed that the sense of direction is sometimes very suddenly disarranged in old and feeble persons. Why this is an interesting system to work on, I've written down here, because we have a lot of very detailed, very beautiful work in animal models, mainly in rats and mice, that have mapped out the navigation circuit. So it's really an ideal system for translational research across species. Secondly, navigation abilities are really very important for independence and mobility. So if you lose your sense of direction, it will have a direct impact on your mobility, and it can also lead to reduced social interaction. And this is actually one of the psychosocial risk factors for Alzheimer's disease. So what I'm going to do in today's talk is I'm going to focus mainly, not exclusively, but mainly on two types of cells that you might be familiar with. These are first place cells in the hippocampus, first discovered in rats in the early 1970s. And what these cells do is that they basically, when you have an animal, say, walk around in this square arena here, and you put an electrode into the hippocampus, and you would then observe cells that have what's called a place field. So they fire at particular locations in this arena. And later on, the Moses in Norway discovered a second type of cells called grid cells, and these are similar to place cells in the sense that they also fire at specific locations. But as you can see here, there's not one, but there's a regular grid of locations where such a grid cell would fire. So originally, these cells were discussed in the context of navigation, so that they kind of give you some kind of position information, like where you are in the environment. But of course, this is also very important information for episodic memory, because when you think of episodic memory basically means a memory for events with a particular spatial-temporal context. So the idea is also that activity in, for example, grid cells and place cells is also used to build up episodic memories. So the function goes beyond just merely navigation. So we do know from many behavioral studies, beautiful behavioral studies, that navigation abilities often show a pretty sharp decline as people get older. And the question is, why is this? What's going on? How does the brain change? What mechanisms could be at play that could explain this? So here's a kind of overview from a review paper where the people like Carol Barnes, for example, have summarized particular neurophysiological changes in the hippocampus, in rodents in this case, that could contribute to changes in navigational behavior or abilities. So we do have things like, for example, synapse loss that has been observed, even though neuronal loss is relatively modest, but you often see quite a marked loss of synapses. You can see when people have demonstrated postsynaptic changes in terms of, say, NMDA receptors. We see neurotransmitter or neuromodulatory changes. For example, GABA processing shows marked declines. And all of this can also contribute then to changes in LTP, so long-term potentiation. So meaning that the aged hippocampus often it's harder to induce LTP. The LTP maintenance is reduced, so it quickly kind of deteriorates. And overall, the whole system, the whole circuit seems to be biased more towards LTD, namely long-term depression. Yeah. So there's a huge amount of evidence kind of documenting these factors. And when you then go into the hippocampus, and let's say you do electrophysiological recordings in an awake behaving animal, you typically observe a number of alterations in older adults. In older rats. So one is shown here, for example, each line here is one spike recorded in hippocampal place cells. And often you see a phenomenon called hyperexcitability, in that those place cells tend to fire much more spikes compared to younger adults. You also see, when you map out different place cells, shown here, and that when you say take an animal and you have 3 cells here, this one fires here, this red one, green one here, blue one there. So you do this over time, you take the animal out, you bring it back into the environment. Usually those cells fire again at the same locations. However, in older animals, this is often no longer the case. You now see here in this example, there's a swap between this red and this blue place field. The green one doesn't even appear anymore in the second one. So place fields seem to become unstable. There's also deficits or changes in what's called experience-dependent place field expansion. And finally, one mechanism that's particularly important for sleep-dependent memory consolidation is what's called replay. Yeah? So that the animal, when you have it, for example, run on a linear track, you record place cells, and firing at different positions on the track. And typically what you observe during sleep is that there is a temporally compressed replay, but that this sequence of, say, blue, red, and green is kind of replayed during sleep. And this replay is often temporally disorganized, and this kind of sequence no longer appears in the correct order, or it's disturbed in other ways. So all of these mechanisms somehow seem to contribute in animals, in aged rodents, to affect and to have a negative impact on their spatial navigation abilities and spatial memory in general. So I want to give you an example here of a study that we have conducted to look at one phenomenon, named hyperexcitability, which is also a neuroimaging study, so functional MRI, in which we basically put people in the scanner and we then show them videos of a virtual environment modeled after a real city. So you see a bird's eye view here of the inner city of Tübingen in southern Germany. This is what subjects saw. And basically what they had to do is they were passively moved throughout this entire environment, and they had to learn the locations of 2 target buildings, like this one up here and this one down here. And then at tests, what happened is that we would position them, say, at this intersection here, and ask them, now try to turn the camera such that you're facing, they're pointing in the direction of this building. However, you cannot see it from your current vantage point. So you have to develop kind of understanding of the spatial layout of this environment to be able to do the task correctly. So this is what a retrieval trial would look like. And here just showing you performance measured as kind of error, angular error. And you see that overall in the beginning, both younger adults shown in black here in this dashed line and older adults shown in this orange line perform pretty much a chance, which would be 90 degrees. But then over time, over multiple sessions, the younger adults quickly get better and they reach relatively low angular error, so meaning a very accurate understanding. On average, older adults, however, really struggle with these tasks, but you can also appreciate here now each line is one participant, and you see that in this older adults group, there's actually quite a bit of variability, and that you have some participants who really stay at very high error levels throughout the entire experiment. But there are also some, like down here, who in the end, even though they might start out at a high level, but in the end also reach levels similar to what younger adults can do. So what are the mechanisms that could drive such performance decline? So when we record, for example, signals in the hippocampus, what we typically observe in younger adults, and this has been shown in many studies, is that initially when the environment is unfamiliar, you see a strong hippocampal recruitment, but as learning progresses, very quickly this signal kind of fades down. And conversely, when you look at structures such as retrosplenian cortex, parieto-occipital sulcus, you see the opposite, in that there's a signal that emerges with learning, and it seems to somehow track the integrity of the spatial map of your cognitive map. In older adults, however, and these are people aged 60 to 80 in this particular experiment, and you see the opposite in that there's a sustained, pretty strong hippocampal signal that does not give rise to learning, and also there's not much change in kind of signal intensity over time in these more posterior occipital regions. So when you then fit a model, like in this case, a dynamic causal model, where you try to set this up as a dynamical system with those two regions here, and then you basically have bidirectional connections between both of them, plus you have inhibitory self-connections to provide some to prevent runaway excitation. What we found in this particular study is that in older adults, only this connection was different compared to younger adults, meaning this kind of inhibitory self-connection within the hippocampus. And most importantly, that this was lowest in those older adults who actually showed the poorest cognitive mapping abilities, so who are kind of very, very much struggle and were not able to pretty much learn anything at all. So of course, from these functional MRI data, we cannot directly infer the underlying physiological mechanisms. We could have something like excitation inhibition imbalance. There could be vascular changes. There could be also degradation of the perforant pathway. But it's quite intriguing to think about reduced inhibition as one mechanism that, as I showed you in rats, could, for example, be related to a change in GABAergic inhibitory signaling that could be causing, or could at least be contributing to those impaired navigational abilities. Yeah, so talked about the hippocampus now. You see hippocampus subfields shown here in yellow. But of course, when you look at the neuroanatomy, we know that a lot of input into the hippocampus comes from the superficial layers of entorhinal cortex. So in particular, layers 2 and 3 that project to various subfields in the hippocampus, like dentate gyrus here, CA3, but also some projections to subfield CA1. So I have already said that in entorhinal cortex, we have those grid cells, so cells that kind of fire in these regular intervals. And I want to show you some cross-species data from what happens to this grid cell, like grid cell signaling in old age. So again, I want to show you some data from another imaging study. And of course, you might wonder, well, okay, but with fMRI, you just have this kind of indirect signal. You're kind of measuring changes in blood oxygenation. So how can this tell you something about grid cells, which is like a cellular phenomenon? So I want to show you like an approach that has been introduced like 15 years ago now, or a little bit more. And basically what happens is the following. Let's say you put a subject in a scanner, and via this mirror here, you present, in this case, a virtual environment, as you can see here. And the subject, again, is somehow moving around in this environment, and they're asked to remember the locations of certain objects here. In addition, you do, for example, T2-weighted imaging that gives you a lot of anatomical detail that allows you to identify both hippocampal subfields, but also entorhinal, transentorhinal regions from these MR images. And then you basically look for a signal that shows what's termed hexadirectional modulation. What does it mean? It basically means, so the underlying idea is that imagine these are firing fields of a grid cell, yeah, and this grid cell is characterized by axes. You see one axis here that crosses these grid vertices, this axis here, that axis here. And the idea is that if you move in this virtual environment, say along one of the axes of this hexadirectional grid, you should see a stronger signal as opposed to when you're moving in between. Yeah. There are some additional assumptions that go into this, but this is kind of the basic logic. So you're basically looking for a signal that shows this kind of 60-degree hexadirectional modulation. Yeah. So this is what we did in this particular study. So identifying entorhinal cortex here on structural MR images, and then we measure the strength of this signal. And overall, what happened is that in younger adults, we could replicate what others had seen before, whereas in older adults, again, those being aged 60 to 82 in this case, there was not really, this didn't seem to be a stable hexadirectional signal in this cohort. We then dug a little bit deeper, and actually what turned out is that, well, there was as a hexadirectional signal, but it turned out to be temporally unstable over time. And this caused these kind of low amplitudes, meaning that you have a grid, but the orientation seems to be changing, seems to be drifting over time. There's no consistent mapping to the environment. So older adults tend to show an unstable anchoring of grid maps to environmental cues. And just, I think 2 or 3 months ago, a nice paper was published that kind of looked at the stability of grid cells in mice running on a linear track. And they also observed lower learning, shown here in aged animals compared to middle-aged and younger animals. And most importantly, they in this study were able to obtain direct recordings of grid cells, and they quantified the stability of those grid maps across multiple sessions. And they saw the same thing that we had seen with fMRI, meaning that the stability of those grid maps was much reduced in those aged animals. Yeah? So this kind of idea that you somehow need to bind environmental cues to your internal estimate of position to allow us to have like stable representations seems to be compromised in older entorhinal cortices here across species. Okay, so far so good, but we have to keep in mind that obviously, and as Jetro already showed, and there are widespread changes in the brain beyond, of course, hippocampus and entorhinal cortex. And obviously, many sensory systems also feed feed important signals into those structures. And I want to give you 2 examples here. So when you look, for example, in the inner ear, where we know, where obviously we have structures such as the semicircular canal or the otolith organ that are crucial for vestibular processing. Yeah, so they sense sense gravity, sense orientation, sense self-motion, and there are quite some marked changes in vestibular processing as people get older. Yeah? And this has to do with things like a loss of hair cells in the inner ear. And this will affect your perception of body motion. Yeah? So how fast, for example, you move through space. Likewise, this study here shows data from visual motion perception tasks. So a subject, for example, would be sitting still and they would be looking at maybe some kind of large screen monitor, and they would be immersed in like an optic flow field. And they're asked to discriminate differences in speed or differences in the direction which you're moving, so heading. And overall, you tend to see that older adults also struggle or are impaired in discriminating speeds and also discriminating directions of movement. Yeah, so it looks like the processing of self-motion cues is also altered, both from the vestibular and also on the visual side. And this obviously is very important information that cells like grid cells use to update their estimate of where you are in the environment. So here's an example of one study that we did with older adults where we looked at that. So as Anne-Sophie mentioned, we do a lot of work also with immersive VR, where people are wearing a headset and we immerse them in some environment and they can physically walk around. And in this particular study, we had them do what's called a path integration task. So path integration basically means, imagine you're here, you're standing here, this is your starting location, and then you're asked to walk along this kind of circuitous trajectory here. And we then stop you at multiple points shown in red here, and we ask you to indicate on a direct kind of beeline where you think your starting location is, even though you can no longer see it, right? So you have to somehow keep track of your position relative to the starting location as you walk along this circuitous trajectory. And we can then measure, first of all, the absolute path integration error. So we can measure, well, how good is someone at doing this? Can they correctly localize this out, or are they somewhat off here? So we can quantify this kind of path integration error. And as you might expect, first of all, it kind of increases over stopping points. So the longer you've walked, the more error-prone your responses are. But likewise, in addition, we also see a nice and reliable age effect. This has been replicated in many studies, that older adults really start to struggle and quickly become more disoriented in these very simple tasks. And I should say these are relatively short paths. So we're talking here just a couple of meters that you're walking. It's not a long distance navigation task, and still people very quickly start to accumulate errors. What's very interesting then is that in this particular study, we then developed a computational model that allowed us to tease apart different error components. As I said, we have sensory signals coming in, we have memory processes, we have response computations. And it turned out that in this study, it was only kind of a parameter that we termed accumulating noise that differentiated between the older and the younger adult group. And then this accumulating noise basically points again to degraded sensory input that then will make your positional estimates more noisy. Yeah, so we have clearly also a sensory contribution here that will then have an impact on higher order spatial computations as we see them, for example, in entorhinal cortex and the hippocampus. Okay, in the final part, I would like to say a couple of words to maybe think about, okay, everything seems to be degrading. There are all these kind of changes that Jetro has talked about. I've talked about changes in LTP, for example. But it turns out, as I've already shown you in this first neuroimaging study, that there seems to be quite a bit of variability as people get older. So these are behavioral data here, or behavioral study, a large-scale study from people in their early 20s all the way up to people in the mid-90s. And this is a navigation task done on a desktop computer. And here on the y-axis, you have total distance traveled. So basically, the more distance you travel, the worse your memory is because they had to find a particular location in a virtual environment. Yeah, so higher values are worse. And of course, as quite as expected, you see kind of overall an increase in these error values as people get older. That's not particularly surprising. But what I would like to point to is that also what seems to increase is the variance, the inter-individual variability. So here in this younger age, most people are still in a relatively narrow performance range. But once we get to, like, say, age 70, 80, all of a sudden the range becomes much bigger. Yeah. And you have some people who are very, very impaired, but you also have some, like, let's say if you take this guy here, whose performance is pretty similar to, I want to say, maybe like 50, mid 40-year-old people. Yeah, so he seems to have a pretty good ability to do this task that is quite similar to people who are about 40 years younger. And this is a very typical finding that that variability in performance, in cognitive performance, increases as people get older. There's a lot of work trying to explain those individual differences in rates of decline in cognitive trajectories. We don't have time to go into that. Of course, you might think of genes and genetic factors that have an influence of that, but of course, at the same time, also environmental factors, lifestyle factors, and there's huge literature on that. This has been linked, obviously, to mechanisms like reserve, so I can build up reserve over the lifespan, but of course also resistance to pathology and potential resilience mechanism. And I'm going to talk a little bit about that now, because there is a literature that you might be aware of on people that have been termed superagers. Yeah? So what are superagers? Superagers are people who are age 80 plus, but who show memory performance similar to 50 to 6-year-olds, and specific and episodic memory. And there was structural MRI work showing that actually superagers tend to show a slower reduction of gray matter volume, in particular in the hippocampus and also in entorhinal cortex. Secondly, there's post-mortem work showing that superagers actually seem to develop less, for example, tau pathology, as you see here, compared to age-matched controls. That for some reason, there seem to be some genetic factors and that the neuronal size is even larger in superagers in entorhinal cortex. And this is post-mortem data, but with tau PET imaging, you can also have use tracers that bind to tau. And you can, and there are a couple of studies, for example, from Bill Jager's lab, that showed very nicely that superagers overall in entorhinal cortex seem to exhibit less tau pathology, and that this the expression of tau pathology is also related to episodic memory decline, meaning those with lower tau pathology also experience lower memory decline. Yeah, and with that, I'd like to finish and just want to summarize, okay, that of course there are multiple mechanisms, morphological, neuromodulatory, that will adversely affect neural coding in the aging brain. There's a strong overlap between memory function and episodic memory, so we'll also see parallel declines in non-spatial, and this we talked about this variability. what's important maybe in a more clinical context is also think about that some of those functions, like red cell functions, also seem to be particularly susceptible to early stages of Alzheimer's disease, for example. And therefore, there's a huge push now to think about to develop behavioral readouts of enteral integrity that could provide, for example, very sensitive indicators of emerging Alzheimer's pathology and disease progression. Yeah. And so keep an eye out for that if you're interested in that. And with that, I'd also like to thank my lab, shown here, current and former lab members, our collaborators, our funders, and thank you for joining this webinar.
Thank you very much, Thomas. That was a great talk. And we have a couple of questions for both of you, Jetro and Thomas, on the Q&A. So I will start right away because we only have 9 minutes.
Should I stop sharing or do I keep sharing?
Yes, thank you. Yeah, so we can see the three of us. So the first question, I think, Jetro, you gave an answer online already, but I still will read it. So from Ziana Yu, can you elaborate on how the right and the left hemisphere differ either structurally or functionally?
Yes, and there's already a preprint out, and I shared that in the chat. Hopefully people can see that, but there's basically the same idea of charting the brain, but that work is also looking into the differences between the left and right hemisphere specifically.
But do you see differences in your work, and if so, when do they emerge?
It's very intriguing. So maybe just a brief answer. So of course we would have the typical development in that preprint, very nicely laid out. But we do see a lot of left-right differences, for example, in association studies where we're looking into environmental exposures and associations to, let's say, left amygdala and left and right amygdala, or left and right hippocampus. So we do see differences in, depending on hemisphere. And it's highly intriguing, but of course, hard to condense.
And so there's a similar question from Paul Winter between then male and females. Do you see differences yourself? Because I know this is something that is quite controversial.
Similarly, I would so much like to include that because, of course, so we have multiple times when, for example, gonadal hormones are influencing brain development and all the development. So one happens during prenatal development, the other one, or the second one, during infancy, and the third one we call puberty. So of course, these are also very key developments or key stages for brain development. And of course, I would probably look first to sex hormones, how they influence differently brain development. And yes, the answer is yes, we do see a lot of, statistically speaking, it's an interaction effect by male or female. It's very common.
Okay. Now maybe more a question for Thomas. We have a question. If sensory input influence structure and function of cortical areas related to navigation, then could we improve navigation if we stream spatial data through a different channel like the skin, sensory simulation or so?
Yeah, very interesting question. Theoretically, yes, and there is work on sensory substitution, for example, in special populations. For example, so in blind adults, and people have tried to use, for example, stimulation on the tongue to develop orientation information or direction information. So theoretically, it's possible. Of course, you then need to think about very carefully about the spatial resolution of the information that you're given. So obviously we as sighted humans, for us vision is particularly important. Yeah. So if the question is, can you use, say, the skin to develop spatial information at the same resolution as you get from vision? Yeah? And that's something that's probably debatable. Yeah. So I know that there are people who have used also kind of just a magnetic belt attached to your waist. And then they try to deliver orientation information by vibrating elements on the waist, but the spatial resolution that you have there is just so much coarser than what you would get from vision. So therefore you cannot probably not achieve the same level of precision there.
Okay, and Philip is actually wondering if this, age-related decline that you see with the fMRI could be linked to some extent to noisy fMRI data or change in hemodynamic or neurovascular coupling, as we do know that those change with aging as well.
Yeah, totally, totally. That's absolutely important to control, to look at these factors in it as well. What I can say, for example, that in this grid cell study, so we look, for example, at temporal SNR in adrenal cortex, we tend to find that it's also reduced in older adults. However, when we regress out those effects, those differences still remain. So it could potentially explain a little bit of the variation, but it doesn't seem to be something that could explain everything away. Second thing is also for neurovascular coupling, when we think of some of those effects, like this kind of hexadirectional effect that I'm showing, it's hard to envisage how exactly a change in neurovascular coupling should affect this particular kind of sinusoidal modulation. It should be more like an overall global effect then. So yes, I'm sure it will contribute, but at least in most of the studies where it's controlled, it doesn't seem to be the full story there.
Okay. And then a question more like for both of you, I would say. Somebody is asking about your thoughts on the role of the blood-brain barrier on neuroplasticity. And I think this is also interesting in the case of development, you know, with the immune system maturing as well in infants. I have young children, so I know how much they get sick. So please, maybe Jetro first and then Thomas.
Yeah, of course. So there are a few measurement types with MRI. Of course, I'm speaking from my area of expertise. It's called magnetic resonance encephalography, MREC for short. And it's highly likely that it's able to capture fluctuations of the lymphatic system, so lymphatic vessels in the brain. And that's highly related to how the blood-brain barrier works. So it can also identify that type of leakages. We wouldn't have as comprehensive lifespan data on that. So it's hard for me to kind of directly comment on that. But it is highly intriguing. And I would guess there are also changes related to overall brain structure and integrity within the brain that then transpires to that. Apart from also diseases, because of course we know blood-brain barrier is affected by many diseases as well.
Yeah, I mean, if I may add, I mean, we know now since a couple of years that most unsaturated fatty acids, they also pass through the brain-blood barrier as if it was not there. So also interesting, the link maybe with metabolism. Thomas?
I don't think I have much to add there. I think you did a very good job.
So then my question to add to that would be indeed, I mean, in the super ager, right? Is there any data on metabolism? Because also throughout the development, right, we know that if we look at least from what I know from the adolescence to aging, right, the metabolism at the entire organism level, but also at the cellular level, only slows down, right? So what could be the impact of that on neuroplasticity?
I'm not aware of any study that would have explicitly looked at metabolic relationships in superagers. But as you said, there's definitely widespread changes in metabolism. I think there's some FDG PET work looking at like glucose. And they definitely seem to show that there are already changes and that those superagers seem to be able to preserve relatively high levels of metabolism. But I think that literature is just emerging. I have to say also that this superager field is kind of growing at the moment, yeah, and it's still relatively young and it's still not to be done. Also, in terms of how exactly do you define superagers, there are very variable criteria that people use. So it's an emerging field, but definitely something that should produce a lot of interesting insights into kind of how to preserve brain plasticity also high into older age.
Do we still have time for one question, maybe?
I think one question, yes.
Okay, then I will ask more from the cellular level. I mean, we know at least from adolescence to adulthood, there's really a change in cellular dynamic at the level of glutamatergic neurons. Like for example, the number of spine, their dynamic changes, it slows down. So how much do you think this might be linked to what you see, right, in this disruption of circuits in aging, Thomas?
So you're talking specifically about things like dendritic spine?
Yeah. Has anybody have looked at what happens to the dynamic and maybe in a excitatory versus inhibitory neuron dynamic in older animals or in older because that's your work tend to suggest that there might be a difference.
Yeah,
with different type
totally.
of neurons.
Yeah, totally. I mean, I have to say that also in the animal field, let's say look at individual differences is also something that's also just emerging. And there is like in the US, there's this big STARS initiative where they do longitudinal trajectories and then try to see, okay, if changes in, for example, spine dynamics could explain that. But that's very rare. That's really very rare. And there's a lot of longitudinal work to be done. Of course, you can do cross-sectional work, but then often it's difficult to pinpoint the exact mechanism that would have caused this. So I'm just trying to think. I think there could be a couple of studies that I looked at, but I'm not entirely sure of the findings. I have to check.
So I guess we ran out of time.
Yes, unfortunately, I would like to thank both of our speakers and our moderator for sharing the recent views and expertise on this topic. I would also like to thank you for attending, welcome you, and invite you to contribute topics at thefens.org/input for the next webinars. And I wish you a very nice weekend.
Yeah,
Thank
you
you
very
for
much.
being here.
Thank you.
Bye-bye.
Thank
Bye.
you.